Can you see my screen?
Yes.
Yes, perfect. How about now?
Yes.
Okay, so I can start. So good afternoon, everyone. My name is Maheshwor Thapa, and I'm postdoc in Sujauli Lab. And today I'm going to present one of the ongoing work from our lab, that is the metabolomics and lipidomics studies of implanting human trophoblast responsive to different microenvironmental nutrients. So let's, oh, sorry, I cannot move this one. I don't know why I cannot change my slide. Okay, I can try. Okay, so sorry for that one. So let's begin with the background information about the trophoblast and then its importance during the implantation. So here, when we look the structure of the blastocysts, the outer surface cells are the trophoblasts, and trophoblasts are the specialized cells that attach and penetrates the endometrium during the implantation of embryo. However, improper invasion of trophoblasts can cause failure in implantation. And it has been widely studied that the uterine microenvironment plays a major role during the implantation. Therefore, we aim to evaluate the potential impact of different uterine environment for trophoblast differentiation and to understand how trophoblast cells respond to different micronutrients. So in order to achieve these objectives, so we performed the in vitro experiment where we differentiated iPSC to tropectoderm cells and then exposed to different nutrient supplements, prolactin, folate, arginine, and resolvin. So for the differentiation, we use differentiation media on day 0, 2, 4, and 6. And on day 6, we achieved the tropectoderm differentiation. Then we supplied the different nutrients and culture for 24 hours before we collect the sample. Next is the sample preparation. So in our lab, we have established two-phase sample preparation workflow for several matrices like plasma, tissue, and cells. And here I am presenting the workflow for mammalian cells, where we have used 150 microliter of methanol for polar phase extraction and 300 microliter of MTBE for non-polar phase extraction. And here we use 100 microliter of water for phase separation. So the non-polar phase extract was used for the lipidomics studies and polar phase was used for the metabolomics studies. And in the result, panel A shows the PCA plot of metabolomics and lipidomics data. And here it shows a clear separation of all nutrient group. Next, we perform ANOVA, and here you can see the significant metabolites illustrated in the heat map. And here we can clearly see the distinct metabolome and lipidome phenotype with the prolactin exposed trophoblasts compared with the other groups. And then we use Momichok for pathway analysis, and it reveals several pathways to be significant. And here, pyrimidine metabolism and aspartate asparagine metabolic pathway are the topmost pathway. Next, we focus on the distinct nutrient phenotype, which is prolactin here. And here we found like several metabolites and pathways, which are the key components of DNA and RNA. And further, we noticed that the pyrimidine metabolism and purine metabolism pathways are the top significant pathway, which in fact are very relevant in aspect of trophoblast differentiation. And in panel C here, you can see the representative significant metabolites. As an example, sulfonatolactate, hydroxyethyl morpholine, they are found to be higher in prolactin group compared with the control. However, uridine, arginine, and oxaloglutarate, they were found to be lower in prolactin group compared with the control. In summary, the prolactin exposed trophoblasts were found to have distinct metabolic profile. So we focus more on the prolactin supplement. Therefore, we plan to perform the prolactin receptor knockout experiments and then integrate the metabolomics data with transcriptomics. And we just started working on the lipidomics, so the methodology and annotations are ongoing. So any suggestions will be highly appreciated. So now I would like to thank the funding agency NIH and the Jackson Laboratory for the institution support, and also many thanks to all the lab members from our lab as well as from Paul Rosson lab. Thank you.